you doing? But you're going to finally see my vaccine results.
It's wonderful to see so many people here tonight. It's really a great pleasure to have all of you here to hear our speaker talk about a topic that's of very great importance to all of us. I don't need to say that. I think that's undoubtedly part of the reason why the crowd is so large this evening. Before I introduce tonight's speaker, though, I'd like to give thank yous to those who helped put this on. Let me just introduce myself first. I'm Bob Einstein. I represent the Santa Fe Alliance for Science.
Excuse me.
I'd like to also introduce Susan McIntosh, who's here in the back, who's the co-organizer of the Santa Fe Alliance for Science, and also the president of providing this evening's refreshments. So thank you very much, Susan, for that. Then I'd like to introduce Ginger Richardson here on my left from the Santa Fe Institute, a co-sponsor of the Santa Fe Science Cafe for Young Thinkers. Ellen Levy is in the back there. She is from the Santa Fe Public Schools. She's the science coordinator there, another sponsor of tonight's event. I don't know if there's anybody here from the Public Education Department from New Mexico PED, but they also are a co-sponsor.
I don't want to hear it.
Then last, I'd like to introduce two people from the O'Keeffe Museum who graciously provide the space for us to have the event. I don't know if Shannon Bay is still here, but she is one of the individuals. Jackie M. is another, and Irina Zerkin is here somewhere, and she wants to make a brief announcement about a program here at the virus. She does her work at Los Alamos National Laboratory principally, but she also has a visiting faculty appointment at the Santa Fe Institute. Bette and I have known each other for a few years, and we're pretty good friends, so it's a special pleasure to welcome her tonight. Thank you very much.
Thank you. Thank you, Bob.
By the way, the only thing I can offer people standing in terms of seating is up here in the front. There is some room up there. Okay.
So I am so excited to see all of you guys. I can't believe the turnout. It's just wonderful that everybody came to learn about this. This is a hard topic to talk about, and it's not the most cheerful way to spend an evening because we're not there yet with the vaccine, and a lot of people in our world are not well because of this virus, but I'm really, really thrilled that you guys want to learn about it. And I want to encourage you to interrupt me because I slip into jargon. I'm a biologist and we speak our own language, and so if you get confused, just stop me. If you have a question or if you have a comment or opinion, I'd really welcome it. It's much more fun for me if it's a dialogue than a monologue, so everybody feel free to ask any questions or make any comments that occur to you while we go. Okay, if I could have the first slide. I'm wondering, could we turn down the lights just a little bit? With that? Thank you. Okay. This is just to remind you about why this is an issue. This virus in our nation has really gotten to be less of a problem because we have very good therapeutics that people can take. But they're very expensive. Not everybody can take them because they have some side effects. You have to take them for your full life. You have to take them every day. You can develop drug resistance. So if you don't take your medicine every day, you can get resistant viruses that no longer have the medicine work for it. So it's really helped in our country, and a lot of people I've known have had much fuller and much healthier lives because they have HIV, and they've been able to get medication. But 40 million people are infected with HIV in our world, and most of those people can't afford any therapy. They're living in the developing world, they're living in Africa and China. So if you're in the US or Europe, you can get therapy, or in Japan, but if you're in China or Africa or parts of South America, so that's really one huge issue and one of the reasons people are getting infected more and more rapidly. 4.3 million new infections happened last year, and about 3 million people died of AIDS last year. There are 15 million orphans now living mostly in Africa. In the US, we had about 40,000 new infections last year, and we have about 1 million of our citizens infected. In New Mexico, we're a pretty small state. We have about 3,000, a little more than 3,000 people living with HIV, and close to 2,000 of these people have AIDS. And in our state, we've lost about 2,000 people to AIDS. Some African nations, like South Africa and Botswana, where the epidemic is really the worst, have about 25% of their adult population infected. And these guys don't all have access to drugs by any means, really only a small number. So when you think about what that means that they're probably going to die in about 10 years. They're usually young people. People get infected in their early 20s and their late teens, and they don't really get to live much beyond 30. And to kind of think about what this might mean, I've been working with people in South Africa for years now, and my colleagues work in pregnancy clinics because I work a little bit with mother-infant transmission of HIV. And in the 90s, those pregnancy clinics would have about something like 2 or 3% of the women coming in positive that were pregnant. And now those same clinics can have up to 50% of the women coming in positive. That means 50% of these young mothers are not going to get to raise their children because they're not going to make it. About a third of the babies, if they don't get therapy, will become HIV positive too and have a very short, very hard life if they don't get therapy. And everybody thinks about what the problem's like in Africa, but they don't think so much about Asia. But it's a really growing, serious problem in Asia too. And India, because it has such a huge population, even though it only has 1% prevalence, that prevalence means how many people are infected proportionately in the population, it still has the largest number of HIV-infected people, and it's still really swamping the hospitals and swamping the healthcare system, and it's rapidly getting worse. So it's a very serious threat to our globe, actually. If I could have the next slide. No, why don't we go back one just for a second, and I'll introduce you to the culprit. So this is a very sick T cell, and the little blue bits are viruses that it's spitting out. So what happens when you have a viral infection, just in general, is the virus binds to the cell, and it dumps its genetic material. It's got RNA, but that gets converted into DNA. Its DNA becomes part of our DNA, so it takes over the cell, and it turns the cell it's infected really into a virus production factory, and that cell just produces massive and massive amounts of virus. And so this is a very sick cell producing lots of virus, and so the goal of the immune system is to kill those guys. Once a cell's become infected, you don't want it anymore. You want to eliminate it. So that's what one of the things your immune system does for you. With HIV, it actually attacks the immune system. So those are the cells it infects. Every virus has its own kind of cell it can infect, and HIV happens to infect immune cells. And so what happens after you've gotten infected is you have some years of pretty good health, but your immune system gets weaker and weaker and weaker. And finally, you can't really fight any infection very well. And so you start getting opportunistic infections, they're called. And when you die of AIDS, that's really what kills you. It says other infections that your immune system isn't able to fight because HIV has really decimated your defense system. Okay, next slide. So the first thing I'm going to try to introduce you to is really the history of HIV and what we know about it and what we learn about it in our laboratory at Los Alamos using evolutionary tools. And so we study evolution, and that's how we grow to understand this virus. It's very important to us. But just so you have the background, this virus was first discovered in 1981 as a disease. So it happened as young gay men living in Los Angeles started to get sick, and their immune systems were failing, and they were getting all kinds of awful things, cancers and pneumonias. They weren't able to heal themselves. And people didn't know what this was. They just knew that they had an acquired immunodeficiency syndrome, so that's what AIDS stands for. And they didn't know what caused it. They suspected it might be a virus, but they didn't know. And people started working very hard, and within 2 years, they had the virus. They had HIV isolated. And so everyone was optimistic. Oh, in a year or two, we'll have a vaccine. We've got the virus. What we did get immediately was diagnostics. So once you had the virus, you could start making tests to test for that virus, and people could find out if they were positive or not. And you could start doing AIDS education because people learned that it was a sexually transmitted disease, so they could start talking about condoms, safe sex, ways to keep the virus from getting transmitted. And so those two things are very useful tools that just knowing what caused the disease enabled us to do. If I could have the next. By 1986, once we had those diagnostic kits, we were able to find it in Africa. We were able to see that it was really an older thing, that we just hadn't discovered it yet, that it had coexisted with us human beings for quite a long time, and that it was really very variable. more variable than anyone had ever seen before in a virus. And the man who was my advisor at Los Alamos when I was a postdoc and first got there was a guy named Jerry Myers, and he realized how extraordinary this variation was. And he thought, this is a major problem. So he went to the NIH and he said, can I start a database so I can gather all of the sequence data from all over the world? We can see how variable the DNA is and then how variable the proteins are as a consequence of that. And we can start thinking about a vaccine. And so he pulled together the first pathogen database. Now there are dozens and dozens of these pathogen databases for different kinds of pathogens. But Jerry really was, I think, a leader in this and sort of invented the concept. And so this all happened at Los Alamos. It took us about 15 years to get drugs that worked. And these 15 years are pretty painful to me because people my age had a lot of HIV-positive friends. And there's nothing you could do for them but watch them get sick and die. And it was very, very hard. I lost a few people I love very much to HIV prior to 1997. But then 1997 hit, and you could give people therapy. So that was a great advance. Next. So what's next is 2007. What are we still working on? We still don't have the vaccine. We don't have universal access to drugs. So 40 million people have HIV, and hardly a very small fraction of those people have access to the therapy that helps so much. Microbicides are things that people are working on very hard, but we haven't got one of those yet either. And that would be something like an ointment or something that you could use that would kill virus. So if you were having sex, you could use that and it would prevent transmission. So people are working hard on these microbicide concepts too. But we haven't got any of those yet. Meanwhile, what do we have? Well, what we have is 40 million people infected. And now the envelope of this virus, if you line up the DNA sequences, has almost 40% variation. So almost 40% of the DNA bits don't match. And you were trying to make a vaccine to that, and a vaccine has to work against all of those variants, and that's really tricky. Next slide. So one of the things that we've been able to use phylogenetics for is to try to reconstruct this history. So this is sort of a black box. We don't really have samples. We don't really have people you can talk to about what HIV was doing at that time. All we have is we know it was discovered in 1981, and we knew that it had been around for a while at the point we discovered it because it was already really diverse in Africa. So we've tried to use modeling and evolutionary biology to puzzle out what happened during this whole black box period. Could I have the next slide? So what could we surmise if we didn't have phylogeny? And phylogeny is just a way of organizing sequences so you can trace their evolutionary histories, and it's very much like a family tree. So if you guys have anybody who does family trees in your family, you've got your mother and your father, and then their sisters and brothers and all your cousins, and the more closely related people you can kind of identify through the tree. And I'm going to show you trees like that, but they're based on DNA sequences of a virus instead of knowing who is grandma and grandpa, etc. So anyway, what could we surmise if we didn't even have any phylogenetic information? Well, there's a few things we can know about this period prior to 1981. Next slide. First off, we knew by 1985 that HIV was extremely diverse, the most diverse virus we had seen to this point in Africa, and that this was going to be a huge challenge. So you just sequence it and you can tell they're really different. This is going to be bad news. Okay, next slide. We knew that the very first positive sample was from the Democratic Republic of the Congo, and that it was sampled in 1959. Next slide. And the reason we knew this is there were stored samples that were frozen from people studying other diseases in the Congo. And so people could go back to those stored frozen samples and say, are any of them HIV positive? And one of them was. What else we know, and we can know this without any other information, is that many, many primates in Africa carry a cousin of HIV. So it's all over the animal kingdom, the monkey primate kingdom in Africa. And that we know that SIVs are very common in bushmeat. So in Africa, in parts of Africa, people eat monkeys the way we eat beef and or elk or something wild caught. And it's very comfortable to them. It doesn't, it's not, oh, monkeys, it's, yay, monkey. So they can go to the markets and buy it. And if you go to those same markets and you test for SIV, which is the cousin of HIV, you find that a lot of this meat has SIV in it. So that's probably how it moved across into people, and I'll talk more about that in a minute. So we could know this without much other science than just sampling. Next slide. The other thing that we could know is that there were big changes in Africa. So people say, why now? You know, if all of these monkeys in Africa had SIVs and people have been eating monkey meat forever, why this century, why our lifetime that HIV decides to emerge in the human population and cause all this trouble? Well, there were huge changes in Africa in the last century. There was needle reuse programs where people were trying to do good, and they'd have one needle and they'd vaccinate everybody against something with the same needle over and over and over again. There was urbanization, so people moved from little villages into cities just like everywhere else. There was forced relocation. There were roads that gave access to places that couldn't communicate before. There were a lot of wars. There was a lot of deforestation and reaching deeper and deeper into the jungles that really had been virgin territory before. So there was a lot of things that changed that could have changed the dynamics, allowing the epidemiology of this virus and the spread of this virus. That's just an example of one of the changes allowing people to move around. Next slide. So if you add to this history what you can do with evolution and what you can do with phylogenies, which are these big family trees of viruses, you can really fill in this history a lot more, and you can also use this kind of information to help build a vaccine. And I'm going to talk about both of these issues. So next slide. So this is now a sequence of DNA, and if you guys have had biology, you know that DNA has got G, C, T, A in it over and over and over again, and that's our genetic code. If you line these things up, you can see that there's some differences. So the red parts are differences. There's 3 differences between those 2 sequences, and that's what you observe. Meanwhile, HIV has got 10,000 of these bases all in a row, but, and we have many, many more than that. But that's not the end of the story, because there's a lot of different ways you might be able to get this kind of mutational pattern. And what we do, what my friend Will and Marcus and I do, I've got some colleagues in the audience, is we try to reconstruct these things, and we try to use our knowledge about the way evolution works to get the best model of the history of these viruses. So the next slide. So you could have had just an imaginary history that illustrates lots of different kinds of change you might have. You could have had a lot of ways to get to these 2 endpoints. If this was the parental strain, it's exactly the same as that parental strain. You might have had just a single base substitution, and that's usually how people think of evolution working. You might have had multiple changes. So to get from G to A, you might have had to go through another step, another base. That we don't have this information because we've only got the present virus, we don't have the past virus. You might have had coincident change to different bases, so it might have been 2 changes to give rise to 1 difference. You might have had parallel evolution, where both of these things changed, but it looks just like a T in both of our contemporary sequences. You might have had convergence, where it goes from C to G, but in this case, it had to pass through another base. And you might have back mutation, where it went from C to A and back to C by the time you could sample it. So we try to use math, and we try to use what we know about evolution to model probable distances in evolutionary trajectories and to assemble these things into these big trees. So there can be a lot hidden, and all we can sample is the contemporary virus, right? We can't go into the past and get much from the past. Next slide. So this is just a slide that kind of shows you visually what a tree might look like, and it shows that lineages have patterns of shared substitution. So you start with some ancestral state, and you get a base mutation, which is just indicated by a color here, and you keep accumulating those mutations, and more closely related viruses would have patterns of bases that look more alike. And so this thing is called a clade. It's a group of viruses that are all more related to each other. So this might be like your brothers and sisters, and this might be like your cousins, and those might be like very distant relations or something like that in this tree. Because of except for now, you've got to think of a virus with a very fast mutation rate. So lineages just have shared patterns of substitution, and you can create these trees based on those shared patterns of substitution. Next slide. But what we have to do is we have to start with just the contemporary sequences. So next slide. So that would be just starting with this information. Then you cluster everything, you try to get the things that are most closely related combined together, and then you start trying to model backwards. And so next slide. You have to recreate all of this with your best modeling ability. And so you can do things like you can reconstruct a probable ancestral state, and I can reconstruct a virus that would have been, say, the eve of HIV or the earliest HIV virus to start this pandemic, and that's one of the things that we do. You can also get branch links, which is an estimate of how long these things have evolved. You can translate that to time, and you can try to get timing estimates. So I'll show you those things in the next slide. This now is one of those phylogenetic trees, but it's drawn in a different way. It's drawn so that it's just kind of all out in a big bush. And each one of these tips represents a sequence. And now this isn't HIV. This is all the primates that have been sampled in Africa. And they all carry different SIVs. And they're all very distantly related because these viruses have been in these monkeys for time immemorial, since they've been co-evolving with the animals. There's a couple of different viruses that have moved into humans from these primates. There's HIV, and the M group is the one that's causing the 40 million infections today, that little bit. There's also something called HIV-2, and that is a cousin of HIV that also causes AIDS, that's found in Western Africa, primarily and a little bit in India. And there's just been an amazing detective story that's unfolded about how HIV emerged from these different primate species. And it's been a series of many, many studies over the past 10 years. And it's kind of fun because the sort of lab rat nerd types who do the sequencing and the phylogenies are working with the primatologists, people like Jane Goodall, in the field who go out and collect samples for them. And they figured out how to do this together. And so the people who study gorillas and chimpanzees and sooty mangabeys in the forest have figured out that if they, like through the chimps, for example, they like to nest and sleep in a tree, so they'll put an umbrella under the tree and catch the urine. They'll put the urine in some dry ice and they'll ship it to Alabama, where they'll get the virus out of that and sequence it. And then they're able to tell, yeah, the chimp was infected. And you're able to get the DNA sequence out and you're able to put it in this tree. So basically, that's kind of how they're working this stuff out. It's actually a pretty beautiful collaboration, I think, and it's been really hard to work out those methods, too. It's not easy. But anyway, there's just been a very recent discovery that brought gorilla into the picture. So if I could have the next slide. Do you guys have any questions so far? No comments? OK, then we'll just keep going. These are beautiful portraits of some of the animals that carry these SIV viruses that are related to HIV. And so you can see many, many different kinds of monkeys carry these viruses. And again, if you go to the meat markets, you'll find SIV in most of these animals if you buy the monkey meat from the meat market. And there was a review in 2005 that had that quote in it. There are no fewer than 36 different non-human primate species in Africa. So next slide. So what happened? Well, we can trace it pretty clearly that this chimpanzee got viruses from these other two monkeys, and they came into human beings to cause HIV. And the phylogeny enables us to do that, the study of the evolution. Next slide. We know that HIV-2 came from sooty mangabe. And I made this slide about a year and a half ago, and we didn't know if there was other monkey species that are contributing other HIV-like viruses to humans. But in fact, if I could have the next slide, in the last year it's been shown that gorillas have a virus that's very closely related to an HIV found in humans in Africa called the O group for the outlier or other viruses, which is a little bit different from the M group. So in fact, gorillas are also in this tight family of viruses that are all sort of quite closely related relative to this whole spectrum of things found in Africa.
Do all these animals get sick the same way people do?
No, they've probably been living with these viruses for a long, long time, and in fact, they don't get sick. If you put a sooty mangabe virus into a macaque, which is an Asian primate, Asian primates and New World South American primates don't carry these viruses. If you put SIV into a sooty mangabe, the sooty mangabe strain into a macaque, macaques get very sick, so it's like HIV in humans. So you can't, there is a model for AIDS in these macaques, and that's what people use for vaccine studies, because it's a way we can model and see if you can prevent disease. But it's not really a very good model for HIV, but it's at least, it's the only model we really have. So they stay healthy.
How different is HIV from SIV?
They're really different. They can be more like 40–50% apart in envelope, but you can align their Pol genes, which is the protein that copies the RNA to DNA. You can align those much more readily, and those are closer. So it really depends on what part of the genome that you're in, and they evolve a little bit differently too. The envelope evolves very rapidly in HIV cases, and it doesn't evolve so rapidly in some of the SIV cases. So they're distant, but they're clearly related. They have the same pattern of genes. You can see that they're the same viruses. And these guys, the chimp ones, are really rather close to the M group case for the HIV. So it's pretty clear that's where it came from. Next slide. So I talked to a lot of people about this. I talked to everybody I can. I talked to cab drivers and people on buses and airplanes. I drive them all crazy. But people get weird notions about why, those Africans, what did they do that gave us HIV? Did they have sex with monkeys? Did they do this? Did they do that? They could have very easily had the virus transmitted just through blood contact. And this is just a person butchering meat, and this person happens to have an injury on his hand. And you can really imagine how a virus might make a leap across species in that kind of a setting. And that could happen to us as well if we were in a setting where we were working with an animal and we were a butcher, or we were a farmer that had animals, or we were a hunter. That could happen. So this just to remind you that it's sort of natural. We're part of this whole life cycle. And this kind of cross-species jump is common. So if I could have the next slide. And just as an example of how common it can be, this is just some of the, zoonosis is the word for that, when you get a hop across species. So you take a virus or a bacteria from one species and you have it make the leap into humans. We've had that happen quite a bit in New Mexico. We have plague for many years now, and we have about 6 cases a year. And in New Mexico, we've had 32 fatalities out of about 250 cases due to plague. And plague usually comes from rock squirrel, and it comes from fleas, basically. And so if you go camping or hiking, or you have mice in your house or something, you might actually get plague from that. Or if you handled a dead animal that you found, that would be another way that you might get plague if you picked up a dead rodent or something. Bad idea to do in New Mexico. We have a new virus, West Nile virus, at least new to the United States. It's an older virus that was discovered in Africa in the 30s. But it didn't move into the United States till 1999, and then it moved to the East Coast. It didn't get to us in New Mexico till 2003. But in 2003, there were actually 202 cases, human cases of West Nile virus. It's in a lot of our birds. So I don't know if you guys remember this, but we were living in Nambe, and there were a lot of magpies, and suddenly there were no magpies, and it was West Nile virus. The crows and the ravens were really decimated, as well as many other birds. But this also can move into humans, and it comes through mosquito bites. So using mosquito repellent is a very good idea in New Mexico. By 2006, we've only had 8 cases. So this is a virus that causes a sweep, and then it kind of diminishes in terms of the way the epidemiology goes, unlike HIV, which just expands. We have another zoonosis that's a very famous one in New Mexico, the hantavirus. And it was first discovered in 1993, but like HIV, it's been with human beings for a long time. What it took was a cluster of cases of people who died with just the same symptoms at the same time, so doctors could recognize it as a disease. So people were getting a terrible, fatal pulmonary disease in the Navajo Nation. They were Navajo people, and they were healthy young people. And people couldn't figure out what it was, but they suspected it might be a virus. And people got on this very quickly, and very soon they discovered that they had antibodies to something called hantavirus. Hantavirus was known in Asia to cause a very different kind of disease. It wasn't a respiratory disease. So at first people were a little reluctant to believe it could be hantavirus, but in fact it was. And the respiratory form is something that we discovered here in New Mexico, unfortunately. And we're still getting hantavirus cases. So far we've had 75 cases, and 31 have been fatal. It's a very fatal disease. Yes?
Sorry, never mind. I read this.
Oh, you could read it. Okay, that's fine. So anyway, this kind of zoonosis is not uncommon, and it's part of being alive. that you're at risk for this. Hantavirus, the way you get that one is by breathing places where mice have urinated, or there's little mouse pellets. You just breathe and the virus is aerosolized and you pick it up from that. And actually, why we had such a bad season in 1993 was it was a really rainy year, and so everything was very lush, and so those deer mice did exceedingly well that year. They were 10 times more than usual. And they were collecting the pinny nuts as we were collecting the pinny nuts. And so under the same trees, people wound up picking up this virus. And that's why we're able to see it as a little cluster. Yes?
How long after the pellets or urine sorry.
After you've gotten infected?
Yeah, how long does the virus live? Can it live in the air?
Oh, I'm not sure of the answer to that. There's actually, where I got these statistics is from the New Mexico Department of Health. And they've got really great sites on all of these different diseases where you can read more about them if you want to. Yes?
How do you think global warming has affected the spread of these infections?
Global warming. I'm not sure. I don't know how to answer that question at all. It's not that I don't believe that it might have. But you might have things like a monsoon. This was actually El Nino. This mouse season was an El Nino season that had a lot of rain. But I don't know if that was global warming or if that particular season was a fluctuation. I certainly think that it might have impact on this kind of thing in the long run, but I can't say specifically. Next slide. Okay, so back to HIV. What's another thing phylogenetics could tell us and the evolution of the viruses? Well, we knew we had a sample that was positive from 1959, and what we knew about that sample was it was a Bantu man, and he lived in a place that used to be called Leopoldville. It's now called Kinshasa. And they had 1,200 stored samples, and this was the only positive one. But there had been a lot of samples that people said were the oldest HIV sample known that turned out to just be a contamination from modern samples. And you could tell that from a phylogenetic tree. So this tree was back in 1997 when the sample was discovered. And we could see that this 1959 thing was close to the heart of the tree. It wasn't like a modern sample way out here. It hadn't evolved so much. So that was very good support that this actually was a very old sequence, that it wasn't like the modern sequences. And so that really confirmed the legitimacy of that sample. So we really did strongly suspect that it was from 1959. Next slide. Okay, so now we've got some evolutionary information we can add to this timeline. So we have not only that AIDS was first defined in 1981 and it's very diverse, but next slide. We know that it came from chimp or gorilla. Now we've got one introduction coming from gorilla. So it's leapt from primate to human at least 3 times, this HIV-1 has, and twice from chimp and once from human. Next slide. We know that the 1959 sample really did come from 1959, that it's the real thing. So we know someone was infected then. Another thing, that's okay, go ahead. Another thing that we can do is we can use these phylogenetic trees, and now here's another phylogenetic tree of the whole sampling of the M group, which is the group of viruses that infects the whole world. We had sampling over 20 years, and this is back in the year 2000, and we used the samples over those 20 years to try to figure out how long these branches were and how long they had evolved from the origin. And we were able to get a timing estimate that this virus began to expand in the human population right around 1930. So this virus was with us at least from that time, at least that's what the modeling suggests. And since our first publication of this result in 2000, there have been several other validations of this result with more data and new methods. So we keep coming up with the year 1930, so that's pretty solid, I think. Next slide. So if we can move on here, go ahead and click. So now we know that not only did it move from chimp, but it began to expand in humans right around 1930. And that's sort of mysterious because here we know we've got a positive sample in 59. We know this virus has been with human beings for a long time. Why didn't we realize it? Why didn't we see it when it's such a dreadfully lethal virus? Well, I think my belief is that the reason we couldn't see it is because you die of other things. You die of pneumonias. You die of a cancer called Kaposi sarcoma. That cancer is endemic in the Congo. You die of diarrhea. Terrible, terrible diarrhea. And that's often how it manifests in Africa. And there's many, many reasons you might get that. So they're opportunistic infections that are driven by HIV, but they're in the background anyway. People had them anyway. So it was sort of hard to pick this out as a unique and new disease until you had the virus cornered, you had it sampled, you had a diagnostic to tell you who was infected. And once you had that, you could see how common it was in this part of Africa. Next slide. Okay, so the other thing we were able to do is these things, now remember I call groupings of viruses that are genetically related clades. We could tell that these clades came from about the 1960s. So that's when those originated. And in our country, we have almost all B clade infections. So we have a B clade epidemic. Next slide. The final thing that we were able to do with this, and I'm not going to really be able to tell you how, is we were able to use something called coalescent theory to try to use those genetic sequences to try to model how the virus would have been expanding in the human population. And the sequences themselves, sort of the information hidden in those sequences, suggests that it expanded very, very slowly for about the first 40 years. So maybe villages, very little movement, just, a small number of people infected. And then it really got to be exponential, rapid expansion right around 1950, 1960. And so that sort of fits with common sense. Next slide. So now we're able to really fill out this history. Go ahead and hit the next button, because we also now have sort of an explanation for why we didn't see it and why we couldn't sample it until at least about 1960, is because there was a slow expansion and then it became much more rapid. Next slide. Okay, so now where are we with this virus? Well, these again are those evolutionary trees where things are grouped based on the distance between their sequences. And these sequences all came from one city. They came from the city of Kinshasa, which happens to be the same city that guy lived in 1959. And this is an incredibly diverse array of viruses. So each one of these clades is a little bit more conserved, but all of these clades are coexisting now in the same city. This is one of those clades had moved into Europe, and this is a sampling from from Amsterdam. So this is one city in Europe, and it's like one of these clades. If you look at a single person who's infected, HIV evolves in every single infected person. So if you sample 2 people from Santa Fe who have HIV, you'll get quite different sequences, maybe 5–10% apart. So that's because it's escaping the immune system within the person all during the course of their infection, and you get a lot of variation in a single individual. If, on the other hand, you have an infection that just spreads really rapidly in a community, like in Kaliningrad, in 1996, there was a drug user explosive epidemic, and this whole drug user community just got HIV. They got the same virus because they all were passing it to one another without any time for evolution. So there's actually 26 of those little tiny lines in that dot. So this is all drawn on the same scale, and you can kind of get a sense about how HIV has evolved over 80 years to become this monstrosity that we have to deal with for a vaccine. Next slide. This little bit right here is the global influenza epidemic from one season. So it's drawn again on the same scale. It's about the same level of variation that you have in a single person with HIV. And the flu vaccine, if you guys have gotten it, you have to get it the next year or maybe 2 years later or something, because the vaccine has to be changed because this virus is evolving too, and it starts to fail after a couple of years. So you actually have to have a new vaccine in about 2 years from influenza, and you've got this much variability to contend with instead of that much. So we have a whole different scale of problem here, and it's very frustrating, but it's worth continuing to try, I think. Next slide. Okay, so this is, remember I was showing you those clades as groups of related viruses? This just shows you how those clades are distributed through Africa. This is sort of the oldest part of the epidemic. It moved probably from chimp into human in Cameroon, and that's the region around Cameroon. So where it's very old, it's had a long time to diverge. And in southern Africa, there's really a single clade epidemic, primarily C clade, like we have primarily a B clade epidemic. Yeah?
Well, I have a question about the previous slide.
Okay.
Which is, Does that mean that HIV is evolving like much more rapidly than influenza?
It's
Or
evolving
is it-
much differently than influenza. So what happens is they both actually evolve about 1% a year. This is evolving about 1% a year in every person that's got it, but you're infected for 10 years, so it's got a long time to change in you. You clear influenza in a few weeks. So at the population level, this guy's evolving 1% a year, and this guy's evolving 1% a year. But what happens is the whole global epidemic with influenza has got that level of diversity. And then a new one emerges the next season, and there's about 1% different, and then a new one emerges. Whereas HIV, it just keeps getting different in every single person. So in influenza, simultaneously in one season, you have a pretty narrow distribution. Whereas in HIV, simultaneously, you have this mess. Yes?
Can you please say, what's a clay? You keep on mentioning.
A clade is, that the word is Greek for branch, and it's one of these groups. So it's a group of viruses that are more closely related to each other than another group. So this is a clade, this is a clade, this is a clade. So one of the ideas for a vaccine is, well, you know, South Africa, as you just saw on the next slide, is mostly C clade. Stick here, though, for a sec. So you could try to just make a C clade vaccine for South Africa. We're mostly B clade, so you could try to just make a B clade vaccine, and then you don't have to contend with the global diversity. But on the other hand, people travel, you know, soldiers move, immigrants come in, people go on vacation. A lot of Africa has trouble with wars and soldiers on the move all the time. So these things, you know, if you make a single clade for a single country, you might have some protection, but it may not last. Yes?
By seeing how it's evolved in the past, can you predict how it's going to evolve in the future?
We can know it's going to get worse, I think. So that much we can say. And there's like been tiny opportunities we've had that we've maybe missed in a sense. For example, that Kaliningrad point source, there's something like that in Thailand, and all of the viruses in Thailand were very closely related to each other for about 5 or 10 years. And people had acted really fast and got a Thai virus, and they might have been able to do something about that. But here, I think our best projection is it's going to get worse. Plus, HIV has other tricks up its sleeve. It doesn't just mutate by base changes like we looked at. It also mutates by recombination. So if you get infected with 2 different clades, which you can, that happens all the time, the viruses start recombining, and then you get this mishmash. And so that adds to the whole diversity picture. Yeah?
So the virus on, say, the very left-hand side of this diagram, it has a lot of diversity compared to viruses on the very right-hand side of the diagram. Why are they all considered AIDS? What actually is it about?
Well, for one thing, they all cause immunodeficiency. They all have the same genes. Part of what it is to be HIV is to be evolved, to be rapidly evolving. So it's enabled itself to evade the immune system. And it has certain bits that are conserved. So if you're trying to align these DNA sequences, there are bits that are relatively conserved, and you can kind of focus on those. And then you'll see variable bits that are exposed to the immune system interleaved in these. And so it's very clearly HIV. It's much closer to other HIVs than anything else. But it's still within the context of being this virus remarkably diverse. And it turns out, you know, now we've been studying other viruses that are just as diverse. HCV, for example, is just as diverse as HIV. But it's all HCV. It all causes the same, you know, issues with liver. It has the same symptoms, manifestations, but it's really a variable virus. It doesn't have HIV's tricks of recombination and things like that, but it evolves really rapidly to point mutation level. Okay, so let's move on. Okay, so we've talked about this, how you might do a single clade vaccine, and that's one strategy, but in our group, we're really aiming for a global vaccine. We're hoping for a global vaccine, because it's also going to not be very cost effective. You can't make a vaccine for every country. And in countries like this, it's completely, you know, a single clade vaccine is completely useless. And if you go to the immigrant population in New York City, the HIV clade distribution looks sort of like that. So there's many, many groups even in our country. Next slide. So, I'm sorry, what was that? Oh, okay, you guys, thank you. So this is just a picture of what can happen if a person is co-infected. These clades all get a letter designation, so they're called A, B, C, D, and then all of them together are called the M group, or the main group. So if you got infected with an A clade and a G clade virus, and they managed to infect the same cell and managed to infect your body, both, then they start recombining, and you can get a pattern of recombination that looks something like this. And this particular recombinant, CRFO2, it's called circulating recombinant form 2, because it was the second one found, is really a dominant form in West Africa, so something about this strain was particularly fit. So it seeded a whole epidemic itself, and then it started to vary from this recombinant base. So that's another way HIV evolves. Next slide. Okay, so there's things you can see about the clades that are sort of interesting, where they have different patterns of evolution and diversity in them. And that's one of the things that we look at. So like this is the envelope protein. This is a picture of the protein. It's been crystallized, and we know what its shape is. And if you look at the diversity of the envelope protein in the B clade, and you compare that to the diversity in the C clade, you'll see that different regions of the protein are more diverse. So in particular, next slide, in the B clade, this thing called the V3 loop tends to be really diverse. And in the C clade, this bit, it's an alpha helix, next slide, tends to be really diverse. So the red bits mean that it's varying really rapidly in people. And we think that's because the immune response is directed towards these regions differently in these different clades. So they've sort of evolved to show different parts of themselves to the immune system. And then these things escape the immune system, and that's why we can see that image of variability. And this is kind of a satisfying story to us. I've been working on this for a couple of years, and then my friend Nana started working on it with me, and we predicted that this would be an antibody binding domain, and everyone said, no, it isn't, because they've been working with US viruses, which are B clade. But just this, about a month ago, I went to a series of talks at a vaccine meeting, and it turns out that in C clade, this in fact is the place that antibodies bind. So that was sort of a nice unfolding, where these sequence predictions really turn out to be the case when you start looking at things experimentally sometimes. Next slide. Okay, so now to the part where we want to try to make a vaccine. Well, there's a couple of parts of your immune system that we would like to stimulate with this vaccine if we can ever get the thing to work. The first is antibodies. So you guys have probably all heard of antibodies. If you've had vaccine, well, you've all had vaccines probably. And what they do is they stimulate this part of your immune system. So all the working vaccines that we have in existence now are antibody stimulating vaccines. And antibodies are just wonderful things. They can do a lot of benefits, but in particular, we want to get something called a neutralizing antibody. And the AIDS virus, we can imagine being up here. This is a picture of its envelope protein. Again, the structure. This thing binds to a protein on T cells and then infects them and dumps its genetic material and ruins this T cell. And what this is an antibody, and this antibody happens to bind to, it's called the CD4 binding site. The CD4 molecule sits on these T cells, and that's what HIV recognizes to get in. That's the doorway. So if the antibody binds to this region, this envelope can't bind to the T cell and get in. So we know that human beings can make these antibodies. What we don't know is how to get a vaccine to elicit those antibodies. So we know that infected people can make them, but we can't figure out how to open up this part of the virus to allow in antibodies to the vaccine and stimulate these responses. And the beautiful thing about the immune system is it has memory. So you know how if you guys get a vaccine, say to measles or something, then your body remembers the measles imprint. And if you ever contact measles again, what happens is it remembers it, and it responds really fast and really powerfully. So that's how vaccines work, and that's how natural immunity works too, and why you don't get infected with the same thing over and over again. So anyway, I think the big challenge for the HIV field is to figure out how to make these envelopes in such a conformation that they'll stimulate the really good antibodies that prevent infection. So that's one thing everybody's aiming for, and we really haven't achieved yet. but it's a goal. And so that's one part of the immune system that we're trying to figure out how to target. And the other part, if I could have the next slide, are cytotoxic T cells. And HIV is kind of different. We're so desperate for a vaccine that with other pathogens and other vaccines, we usually want to see complete protection, and that's if the vaccine protects you from infection, then it's a good vaccine. With HIV, we may never be able to achieve that, but what we might be able to achieve is something different. You get infected anyway, but your immune system is able to keep the virus in check. So it's able to keep down your viral load. It's able to keep you healthy for a lot longer. It's able to keep you less infectious. So that would be a satisfactory goal for an HIV vaccine. And T cells click in a little bit later than antibodies do. And so people are working very hard for the first time on trying to get a T cell vaccine for HIV. And what T cells do is they recognize infected cells and then they cause them to explode. So they're wonderful, part of your immune system. And the way they do this is that you have a cell, it's infected with a virus, and it becomes a viral production factory, and it's making viral proteins. It chops up your cells will chop up those proteins. And this is called a class 1 molecule. You've got these on every single cell in your body, and we've all got different sets of them, actually. So we all see viruses in different ways. This class 1 molecule will carry a little bit of virus protein. So the red bit is about 9 amino acids worth of viral protein that's been chopped up. It will carry it to the cell surface and present it there. And T cells can see this. They can see a class 1 molecule holding a bit of virus. So what they do, here's now a T cell, and here's now a virally infected cell, and this guy recognizes that this guy is virally infected and sick. So he then sends a bunch of what things called chemokines across that cause this cell to explode. You can see that gaping hole. So that's the aim of a T cell, is to kill virally infected cells. And this is the place where we've been having some hope about our current vaccine work. If I could have the next slide. I'm going to just tell you about one of our older ideas, because it's the one that's gone the farthest and it's also the simplest to explain. And you guys have heard a lot about evolution already tonight, so I'm not going to go into some of the more complicated concepts we have cooking. But this idea was an old idea. I first started trying it in about 1995, and I first talked about it in 1997. And biologists told me I was crazy, because they didn't think it would work. So the idea was this, that you can reconstruct, you can estimate what that ancestral sequence is. So you can use these tools we have to try to get back to what the sequence would have actually been. And not only can we do that, but we can also do something called a consensus, where we take the most common base in every position. And as it turned out, these things were exactly equivalent, the consensus and the ancestral, for the first one of these prototype vaccines that we made. And finally, in 1999, I talked my friend Fong into making one of these things. And you have to synthesize the whole stretch of DNA, and then you have to get it expressed. And then you have to show that it binds antibodies and it folds properly, that it's a well-behaved envelope. This all takes about 2 years. Then you have to start putting it in small animals like rabbits or mice or something and see if they make a good immune response. And they did. And so then the next step is you put it into primates and see if primates make a good immune response. And they did. So far what we know about this is this artificial protein that is an estimate of the ancestral virus is really doing what we want it to do. And if I could have the next slide. This is the test that we devised to try to look at this. So this is our protein and where it sits on the phylogenetic tree. It's called the CONS protein. And this is a B-clade protein that's being used in vaccine trials now. And the way you've traditionally done this is to just take a natural protein, right, and test that natural protein and see if you can make good cross-reactive responses. So this thing, you can see that it's genetically very far from all of these other clades. And this thing is central to all of the clades, right? So the distance in number of DNA changes is about the same from this to all of these guys, whereas this you have to go a massive distance, and the things are really, really different and very difficult to align even. So what we did was I picked out 10 different proteins isolated from all over the world that were contemporary isolates, and those are the red ones. And then we injected a bunch of macaques with this protein or that protein, and we asked which one stimulates immune responses that can react with these guys, and how well can they react. So the next slide shows you how well they react. And I was really very excited about this slide. It's not published yet. We've just got the paper submitted. But every little section here is one of our macaques. And these guys got the natural protein, the clade B protein, and these guys got our central protein. And the red ones are B clade responses, and the other kinds of shades of blue are all these other clades that we tested. So we tested 10 different proteins. And we said, how many responses are we able to get cross-reactive good responses to with this whole slew of proteins? And you can see, I mean, which one would you want? You can just see, right, visually, this one's doing a lot better. So we're pretty excited about this concept. And we're hoping we're going to push this forward into a small human trial to see how it does in humans, hopefully this next year. And meanwhile, we're trying to work on improving this. So the next step that Will and Marcus and I are working on is trying to get this one step further. But this is where we're at so far. The virtue of this thing is that it's a single protein, and it does so much better than a single natural protein. So we're pretty excited about this result. But the caveat in all this is it's just in monkeys, and our immune systems are different. And things that work in monkeys don't always translate to working in humans. So it's a big leap, and you just have to do the test in humans to find out if it's going to be successful. Okay, next slide. Okay, so this is just going to summarize what I've been telling you about. The HIV virus has had about 80 years to expand in the human population. And in those 80 years, it's become an extraordinarily diverse virus. We still don't have an HIV vaccine, and this has been since 1983 that we've been trying. Several good ideas have failed when tested in human trials. One of the human trials was just aborted about 2 weeks ago for failure, unfortunately. But we're optimistic about the central vaccine design concept, this in the middle of the tree concept. And that's just one of many very good ideas that are in the pipeline. So that pipeline, it takes about maybe 5 to 7 years to have an idea go all the way into a human trial for HIV. And it has to pass every test, safety, that it's working all along the way. So there are a lot of things that people are trying that are very clever, that are in intermediate stages, and hopefully some combination of these things will work. So that's where we're aiming for. And if I could have the next slide. Okay, we can just go through this quickly because I already talked about them. Okay, this is just one more reminder of why this all matters. This is a wonderful woman named Priscilla who lives in Cato Manor outside of Durban, South Africa. And one of my neighbors from Los Alamos, a wonderful woman named Maureen Bearclaw, who's a photographer. An artist just got so moved by the AIDS epidemic that she went to South Africa and Uganda and took care of orphans for a while. And she met Priscilla there and helped Priscilla out. The next slide. These are pictures she took. This is Priscilla's house, and it's a little 4-room house. And these are some of the orphans that she takes care of. The other 30 orphans that are in their family are at school at the moment. There's some teenagers there, like you guys, and they're taking care of the little ones, and all of them have lost their moms and dads to AIDS. And Priscilla gets these kids by somebody leaving them in a basket on her door. And her church helps her feed them, and people help her feed them. Marina has raised money for them. But this is just one extended family, and this is happening all over Africa. So we've got to do something. We've got to keep trying. Next slide. This is where I work. I work at the Los Alamos National Laboratory and the Santa Fe Institute. And next, click forward. This is who pays for my work. So the National Institute of Health pays for most of it. Los Alamos itself pays for some of the theory and the work that we've done. In fact, it pays for my 2 colleagues sitting in a room. The Elizabeth Glaser Pediatric AIDS Foundation gave me years of support for the development of the consensus vaccine. And the talk that I've given you this evening really represents the work of dozens and dozens of people. So I've done some of this work, but it's been in collaboration with many wonderful friends and colleagues. So I'm not going to tell you all their names because it wouldn't mean so much to you, but in a scientific talk, you usually have a big list of everybody at the end. But just rest assured, this is the combined effort of many good brains. And it's been an interesting process. I just wish that we were a little bit further along the way. And I'd love to talk more if you guys have more questions. Yes?
Can you please tell us about the so-called cocktail of drugs that was given to the gay community in the mid-80s?
It's still
It
given.
was very effective, and still given, and was very effective in combating the HIV, as opposed to the early years.
Yes, I can tell you, and it's about evolution again. And in fact, understanding evolution is what enabled us to go to the cocktail. So what happened was, early on, people started making drugs that hit the polymerase, which is the thing that copies RNA to DNA. And they target that, and that's a pretty conserved part of the virus. So they had these drugs, but they were trying one at a time. And they would give it to people, and it wouldn't do them much good. So they wouldn't get better. They developed drug resistance really quickly. So they get a mutation that would enable the virus to be resistant to the drug, and they would just become resistant and wouldn't do much good. So then people thought of something called protease inhibitors. Protease is another viral protein that's really conserved, and they tried those one at a time. And there was a particular amino acid that this drug bound to that was preserved all over the world, every single virus sequence. As soon as you gave the drug, within 6 weeks, that amino acid had mutated to something else, and the virus was completely resistant to the drug, and it didn't do any good. So I remember actually there was a guy from Merck came to Los Alamos and talked about that, and we just thought, 6 weeks and you're drug resistant, what are we going to do? This is hopeless. And someone came up with a brilliant idea in 1996 of combining 3 drugs at once. So instead of just giving one at a time, you give 3, and that would mean the virus would simultaneously have to have 3 mutations to escape. And it can't do that. It can do 1 at a time, but it can't do 3 at a time. So that's why the cocktail works, and it works really well. The problems are that it's hard to take 3 drugs for your life, and you can't stop. Because as soon as you stop, the virus wakes back up again. They do have side effects, and some people are bothered by the side effects. Some people can take them pretty well, but others don't. Sometimes they cause nausea. Sometimes they cause a redistribution of fat that makes people look bad, and they feel bad about that, and they don't want to take them. And sometimes you develop drug resistance. So you're taking your 3 drugs, and you get resistance to one of them, so your doctor gives you a new one, and then you just keep having that happen until you're finally resistant to really pretty much the package of what's available now. So they have, is it on the order of about 17 different drugs they can choose from, but they're families of related drugs. And so they try to give you a cocktail that works for you. And actually they sequence the viral genes to see what you might be resistant to when they're giving you that combination. And they work really well. They're very effective. Pardon? I'm sorry, I couldn't hear the question. It was 1996 that it was realized, and they started giving it to people in 1997. And it made a huge difference in people's lives. It pulled some people right back from the brink of death. It was incredible. Yes?
Are the primates immune to the SIV?
I'm sorry, say it again?
Are the primates immune to the SIV?
They're not immune to it. Well, they do have immunity to it, so they make immune responses. They don't clear it, though, and they don't get sick. So,
do you think that humans will create some sort of immunity to it?
to be able to live with it, I think they might. It's possible. It's very hard for me to imagine how much death and mayhem and suffering would be on that path, but I think it's possible. Yes?
Let's say theoretically I had to eat a monkey that might be infected with AIDS, right? Let's say I'm like in the middle of Africa gonna die. Does it like die at a certain temperature that I could just cook it to?
The virus? Yeah, HIV does in fact die at a certain temperature.
So if I cooked,
If you cook the meat well, you'd be all right with HIV. Different things are not so pleasant. The prions, for example,
you know what?
are a little bit more
One day,
dangerous.
One of you guys are gonna be like, you know, you guys are gonna be lost in the middle of an ocean, and you're gonna have like this guy with HIV who's on the boat with you, and he didn't have his medication, he died, and you're gonna have to eat him, and you're gonna say, "Well, I'm glad it."
And you're going to have to build a really big fire on the boat so you can cook him well. Yes. You're going to want him well cooked. You can also wear gloves when you butcher. And in fact, if you guys hunt or if you have family members that hunt, they should probably wear gloves when they butcher. It's not, if they do that, if they cut up an elk or something. Good idea. Yes?
How long can HIV live in a body that's at neutral temperatures that's dead?
Oh, I don't know the answer to that question. He's wanting to know how long HIV lives in can persist in a dead person. So how long is that person infectious? You guys are interested in dead things. I'll tell you a dead thing story that's sort of sad, but do you guys know what Ebola virus is? Have you heard of Ebola? It is a terrible thing, and that virus is really passed through dead bodies. And so someone will die, get it in the community, then people will wash the body and stuff, and everybody who handles the dead body gets Ebola. But for animals, it's the same way. And if they find like a dead gorilla or something, they'll eat it. And the dead ones can carry Ebola sometimes. So that's caused the infections of villages in some cases. Yes?
If every human being on Earth got AIDS, would it eventually like integrate with our DNA? Like, would it eventually become like part?
Oh, yes, it probably would. And in fact, much of our DNA is retrovirus. Lots and lots of our DNA is ancient retroviruses from way back. But it's not from before we were human. As mammals have evolved, we've evolved with retroviruses. So we're loaded with retroviral DNA. There's weird facts like I think there's more retroviral DNA than our DNA in our DNA. There's more bacteria in your body, more bacterial cells in your body than your cells in your body. So what are you exactly? I don't know, but you're some weird mix of retroviruses and bacteria that walks around in the skin. Yeah, yes? Pardon me? There's interesting things about HIV where some people might be immune to it. So she asked if anybody's immune to HIV. And there's some people who have high-risk sex for years and years and years and don't get infected. And those people are really interesting, so people study them a lot. There's a couple of cases where you can study those people. One is a monogamous relationship, and actually the gay community is really fabulous about volunteering, I think, because they've lost a lot of friends and loved ones to HIV. So there's a lot of gay couples that have volunteered, and sometimes you'll have one partner HIV positive and one who's not positive, and they come in and get blood all the time, and people track them for years. And sometimes, very rarely, the other person doesn't get infected. And also there's prostitutes in Africa who maybe have been a prostitute for 10 years and have a clientele that's 25% HIV positive, and they don't get infected. And people are very interested in trying to understand why. And it might be that they just made a really good immune response, and they saw the virus, made a good immune response, killed it, or kept it down to the level you can't detect it in them, and they've gone on and been okay, but they're rare. They're very, very hard to find, these people. There's a few other people who have HIV who don't get sick, but again, they're very, very rare. It's not typical. Yes?
Could we go back to the first slide? There's a lot of information.
Sure. Do you mind taking us back there, Kim? Thank you. I'm not sure. If you guys are interested in reading more about this, the New Mexico Department of Public Health has got great information. Okay, next slide. That one's not the informative one. There we go. Okay. Yeah. Yes.
Like some cancers, it's like intensely painful to have, and you're just in pain 24/7. With most cancers, like stuff like that, or illnesses. And with HIV and AIDS, do you feel like pain, or is it just?
Oh, you can. HIV takes people out in very, very different ways, and I don't know any of them that are pleasant, and it takes them out slowly. So, the friends I've lost to AIDS. One of them had dementia, so a lot of people who have HIV get dementia. He didn't recognize anyone he knew. He weighed maybe about 80 pounds. His mind was gone. The only person he could recognize was his mother for his last 6 months of life. He was a beautiful, brilliant man, and he could feel his mind going, so he had to watch his memory go for about 6 months. He died at 29. Another friend died about 25, and he was just this gorgeous, happy young guy, and his last 2 years of life, he was barely able to move. He was lesions all over his body. He was really thin and gnarled and in a lot of pain. He looked like he was an old man, and HIV has lots of different ways to take people. So it's not, because you die of different diseases. In Africa, you often die of wasting disease, so that's diarrhea. And if you can imagine bad enough diarrhea for months that's going to kill you, that's not going to be fun either. It's not a pretty way to die. Yes?
Just a hypothetical question. If someone were to contract HIV and then, or HIV/AIDS, and were put in a situation where they were exposed to no diseases or viruses or anything, like just a completely sterile condition, how would they be affected?
Well, I think you can't do the experiment, so I think nobody knows. But you also, the virus is also not good for you itself, but it really isn't the thing that kills you. And I just think that it's a hypothetical question that doesn't have an answer, basically. Yeah. Yes?
You said there are drugs that make the infection less infectious. Does that mean that it reduces the risk of being able to spread it, or
So I think it probably does, yeah. So your viral load goes way, way, way down. But it doesn't mean that it's safe to have unsafe sex. But it probably does reduce the transmissibility. It's kind of hard to measure the transmissibility of HIV because you can have sex a lot before you finally transmit. The reason there are 40 million people infected is people have a lot of sex. But it takes a lot of sexual acts to actually transmit the virus. Yes?
Is HIV going to get so bad to the point where we're going to have to accept people out people on HIV?
Yes, that's what we're gonna do.
Let's hope he's not in a position of power. I think the question was, if I heard him correctly, is HIV going to get so bad that we're going to have to kill people with HIV? Well, it's only sexually transmitted, really, or transmitted through drug use, through needles. So it's very I lived with a person with HIV for 5 years, and I was not afraid, and I really understood this virus. It's not something that's casually transmitted. You have to have sex with the person, you have to share a needle, or you have to have a blood transfusion. So you're safe to sit with somebody with AIDS, you're safe to hug somebody with AIDS, you're safe to kiss somebody with AIDS, but not further than that.
Technically, you could have sex with someone
Technically, you could.
with a condom.
Condoms help a lot. They're not perfect, but they really help. They really, really dramatically reduce your chances.
Lived here like in the 80s,
Uh-huh.
and she said that like all of a sudden it was just like this. All of her friends started dying off, and it was like they would get like a flu or like a cold, and then the next week they would get something else, and then they would eventually die of pneumonia or something.
Yeah, pneumonia
And
kills
it
a lot of
started
people with
just
AIDS.
a common cold, and nobody knew what was going on, and they said it was such like massive like loss of people that they were end up burying like 6 people a week.
Wow.
7 people a week, and like they didn't know why, and so like it ended up that everyone just was like, don't even touch me. They didn't know what was going on, so it was scary, I guess.
Well, it's good to understand, it's good to understand what causes something, and it's good to understand what can't transmit it, so you don't have to be afraid of somebody with HIV, because you really don't. There's no reason to be. Yes?
Yes. If a single person wanted to take an HIV test, where would they take it, and how positive would it be?
The tests are really good, and I'm sorry that I can't tell you how to do it. You can go online to find places in Santa Fe, though, if you just go on the web and ask. But actually, something else, I think that teens can get tested without having parental permission if they want to know for any sexually transmitted disease in the state of New Mexico. So it's actually something teens can do.
The initial test would be positive, you didn't have to put it together to get a second opinion.
If I had a positive test, I would there's a fabulous AIDS clinic in Santa Fe, just a wonderful group, and people who are positive often go there. I think I would go there and get a second test if I tested positive, but the tests are really reliable. They're very reliable at this point.
The reporter does have an ad every week giving you a place where you can get a free anonymous HIV test. So.
The thing for teens, I think what the law is in New Mexico, if I understand it correctly, and it's just from reading an article on the web about it, but I think teens are allowed to get tested for sexually transmitted diseases without telling their parents or their parents the results. But if you're positive and you want to get treatment, you can't get treatment without talking to your parents. So I believe that's the law in New Mexico. Other questions? Do you have anything you want to add, Will? Okay.
So the medications, are they very expensive?
They are expensive, yes. I can't think of the cost just off the top of my head, but they do cost quite a bit. It helps to have good medical insurance, I think.
One or two last questions for Bette, Dr. Korber?
Oh, sure. Teacher? Yeah.
Well, I guess some of you are here tonight, and you have to have your slip signed, or you have to have somebody sign, or that you actually
Good
come.
questions. Thank you
So
for
I
picking
would
up.
be glad
Appreciate
to
it.
sign those if you want, or perhaps Dr. Korber would. But in any case, let's please join me in thanking her for a wonderful